In this lecture, we're going to cover translation, which uses mRNA as a template in order to make a protein. So the skills for the exam will be, you must be able to use a DNA sequence to predict a protein sequence. You should be able to list the enzymes needed to complete translation. You're going to describe the roles of tRNA, mRNA, and rRNA in protein synthesis. Protein synthesis means the making of a protein. You're also going to list where translation will occur in both prokaryotic and eukaryotic cells, as is a theme of this class. Okay, so with all these questions, you should really go back and start with the central dogma, which tells us how information flows in the cell. So we can see, here's DNA, and each one of these arrows shows us what is the template and what is the product of each of these steps. The first step uses DNA as a template in DNA replication to make more DNA. So DNA is both the substrate and the product of DNA replication. This occurs in the nucleus. We covered this a couple lectures ago. DNA is also the template to make mRNA through the process of transcription, and I've abbreviated transcription TXN here. So DNA is the template, mRNA is the product. This is done by RNA polymerase. So the polymerase is named for its product, which would be RNA. This occurs in the nucleus because that's where DNA is located, and this results in an mRNA product with a start codon. mRNA then leaves the nucleus and then this goes into the cytoplasm at least in eukaryotes. And in the cytoplasm, this can undergo the process of translation, in which mRNA is used as the template to make a protein product. Okay. And so this, we already know, uses the ribosome. So the ribosome is the polymerase for translation. So this is a polymerase still because a protein is a polymer. So the ribosome is going to make the protein polymer. This isn't protein polymerase, it's called a ribosome. Okay. And this is how the cell gets all of its proteins, is through the ribosome using mRNA as a template. Okay, so we already covered the process of using DNA to make an mRNA strand. So there's two strands of DNA, the complementary strand and the template strand. And just in case you haven't remembered from a little while back, the complementary strand is not used to make the mRNA. The template strand is the information for the RNA. Okay. So these two strands, these two DNA strands, are anti-parallel because one would go from, you would read from left to right, and the second you would read from right to left. So when we set it up in this way, it's actually perfect to look at the mRNA. So the mRNA, again, you read from 5' to 3'. So in this setup, which it will be set up like this on the exam too, so don't worry about that, it's absolutely perfect because we can just read the mRNA directly without having to flip it. And I know you're perfectly capable of that, but let's just not make the exams more complicated than they need to be. Okay. And in the mRNA, just as we did before, the T's will pair with A, and the A's will pair with U's. So what I like to do is I like to go through and do all the U's first. You can do this however you like. It does not matter to me. As long as the answer is correct, that is the only thing that really matters. Okay. So everything else is the same, it's just that instead of pairing with T, mRNA is going to pair A with U. Okay. And also in the last lecture, we were able to identify the start codon. So that's the first thing that you need to do in order to locate where the protein is going to start. you must find the start codon. So find the AUG start codon on the mRNA sequence. Okay. And this is the exact same example that we just did, so we know that the AUG is right here. And it doesn't have to be the first one or the second one or the third one. You just keep looking from left to right until you see 3 letters in a row that are AUG. Okay. And that is where we're going to start translating our protein. Okay. So in this process, what I like to do is, because this is based on codons, series of 3 bases, I like to draw lines every 3 nucleotides, every 3 nucleotides into codons, just to help me see. So 1, 2, 3, 1, 2, 3, 1, 2, 3. Okay, we do not care about anything that happens before the start codon, because remember that the polymerase for RNA, the RNA polymerase, has to start at the promoter, so we're going to have some extra on this 5' end, and that's totally okay. But we start at the start codon. Okay, so 3, we're going to use our codon table in order to figure these things out. So pause the video for a moment, find your codon table. If you cannot find it, print a new one. Okay, so I'm going to teach you really quick how to use a codon table. So the first position is listed here, second position's across the top, and third position's down the side. So what do you think that means? So in our pretend codon, or let's start with the start codon, shall we? Start codon, hold on. Okay. Start codon is AUG. So this goes from 5' to 3'. So we know that this is going to be position 1, this is going to be position 2, this is going to be position 3, always. From left to right, it goes 1, 2, 3. So in the first position, we're going to look in A's. So that means it must be somewhere in the A row, and it's not going to be anywhere else. Okay. Do not write on your codon table. You do not need to. This is just for example. In the second position, we see U. So we've finished the first position, it's here. Second position, here's U. So we ignore everything else. It has to be somewhere here. Somewhere in this box. So we can ignore all of this stuff. Okay. Oops. I overstepped. Because we need this for the next step. Whoop, 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 whoop. Okay. And then in the third position, which we see here on the right, we see G. So that means it's going to be on the bottom here. So, and lo and behold, AUG, it does not say start codon. So that's why you have to memorize it, but we know that this is the start codon. So this right here, MET, is the amino acid that's encoded by the AUG start codon. So we can tell from the first position, second position, third position, AUG. You do see that this is highlighted, and that means that it's special. Something else I want to point out is that UAA, UAG, and UGA are all stop codons. So we have distinct start codon right here at methionine. We also have distinct stops. And that's going to be really important for the protein. You have to tell it where to start and where to stop, because the mRNA sequence is so long. And if we add extra amino acids, that's going to change the structure and change the function. Okay, so our first one, first position, second position, third position, we discovered that this is MET, methionine. This can also be abbreviated capital M. Okay, so the next codon, UUU, on your codon table, see U first position, U second position, U third position is this top line. That's going to encode PHE, phenylalanine. So that's what we're going to write up at the... Okay. So UUU, PHE. So that would be abbreviated F. You'd be given a table for these abbreviations. Next is UAA. Okay, so we've got U first position, A second position, A third position, and we see that UAA encodes stop. So you have two choices here. You can either write the word stop or you can draw a star. Star means stop. Okay, so we use our codon table for each codon. You record the amino acids. And you continue until you reach stop. You reach the stop codon. And then you write a star or the word stop. Okay, so in this case, this is our primary structure of the protein. It starts with an M and it ends with an F. And then the entire complex falls apart. Okay, so next I'm going to draw you really quickly what this process would actually look like in a cell. So, we've used the DNA to make an mRNA sequence. So the mRNA travels from the nucleus into the cytoplasm where this whole process will occur. So this is our mRNA. And remember, it's single-stranded. And this has all of the bases that's going to encode our protein. Okay. Somewhere in the middle here, we're going to have AUG, the start codon. So the role of the mRNA, this carries the codons that carry the message for the protein. When we identify the start codon, there's another type of RNA in the solution that's called the tRNA, because it kind of looks like a T. This tRNA holds the anticodon. So the anticodon base pairs with the mRNA codon. So A base pairs with U, U base pairs with A, G base pairs with C. So you'll notice if you look up UAC on your codon table, this will not encode methionine. That's because the codon table is only for codons. The codons are on the mRNA. Codons are for the codon table. It is not called an anticodon table. It is called a codon table. Okay. So, somewhere in the solution, there are anticodons that are going to pair with that are going to complementary base pair with our lovely, if this will ever move. Oh my goodness. Okay, anyway, so these are going to base pair with the codons. So A base pairs with U, U base pairs with A, C base pairs with G. So that's what that's about. Okay. So this tRNA is going to base pair with the start codon, and that is the first step of translation. Once this has formed, then the ribosomal RNA and the ribosome can assemble around it. So I'm going to draw like this. The ribosomal large subunit has 3 openings. 1, 2, 3. And I'm drawing these every codon. Okay. So, depending on which way that the large subunit, which looks kind of like this, large subunit, ribosome, which is made of ribosomal RNA and proteins. And then at the bottom, to keep the mRNA nice and sandwiched in here, we've got the small subunit, which is made of R, RNA, and proteins. Okay. So, the rRNA makes up the ribosome. The tRNA is going to carry the amino acid, MET, that's encoded by the codons on the mRNA. So mRNA is for codons. The tRNA carries the amino acid, and the rRNA makes up the ribosome. What color haven't I used? This one. rRNA makes up the ribosome. Okay. And then the ribosome will actually make the protein. That's our polymerase here, as I mentioned previously. Okay, so this is the initiation complex. So the tRNA is going to base pair with the start codon, the ribosome assembles around it, and now we're able to move. So the next tRNA will then float in and base pair with the next 3 bases, 1, 2, 3, carrying its amino acid. So whatever this amino acid is. This will then transfer to the next, and then the old tRNA will exit. Okay. And I have a much better video of this whole process coming up, so please be patient.